All right, today's lecture is going to be on looking at Earth's history, the early Earth, the conditions of the early Earth, and then the evolutionary history of life after that point. We're going to talk a little bit about some competing theories originally on how life began, and then talk about what we kind of think is the case now. Then we're going to kind of finish off with human evolution and how humans evolved from a common ancestor that we share with some of the other great apes and some of those other lineages of humans that existed in the past but no longer do, that you probably have never heard of before. So, the universe itself is about 14 billion years old. So we say 14 billion years old. It's actually 13.772 billion years old, but we'll call it 14 billion years old. And it was born from a singularity. This is all in the realm of physics. This is called the Big Bang Theory. And that singularity was a point of nearly infinite energy and density of matter. And then it exploded from that point. We do have a lot of evidence to show that this was the case. We have the cosmic background radiation of the universe, which exists. We have the rate at which the universe is expanding, and almost everything is moving away from each other. So, there's still expansion going on. But because it's a biology class, we don't focus that much on the Big Bang. Now, our sun started to coalesce about 4.6 billion years ago. So our sun, the Earth, our solar system in general formed about 4.6 billion years ago. And it was definitely on Earth not a place that life could exist. It was a hot ball of molten rock, essentially, that was being bombarded constantly by lots and lots of meteors and objects from space. And in fact, at one point, the moon formed from a giant object slamming into the Earth, and then that was the size of like another planet, and the moon broke off of that and coalesced into a kind of a large celestial entity. And what's unique about Earth's moon, it's the largest moon in the solar system, even though the Earth is not even remotely close to being the largest planet. So I think that's kind of an interesting piece of information there. So it took about 400 million years for the crust on the Earth, that molten rock, to finally solidify, and then eventually liquid water formed on the surface. That took a long time. This early Earth, though, had no oxygen gas present whatsoever, and which meant that cellular respiration as a process could not have occurred on the early Earth. And as we talked about at the beginning of the year, remember, water is the key to life. And why is it so important? Remember, it has those solvent properties. It dissolves lots and lots of things in it, so it's a great place for chemical reactions to occur. It also has a really high specific heat, which allows for limiting temperature fluctuations within the within the water. And there are other properties too, the adhesion, cohesion, that kind of thing. But that's more important, I would say, for like land animals and plants and things like that. It also has the insulation effect that when it freezes, at this point there probably wasn't tons of frozen water, but when it freezes, it stays at the top and doesn't sink to the bottom, so plants do not get crushed. Remember, water in its solid form is one of the only solids that actually is less dense than its liquid. So not long after water finally cooled, like it came from the gaseous form and cooled down, life formed, and we have evidence of that in the fossil record. We have fossils with prokaryotic looking life and evidence of prokaryotic life within it at that time. And before there was actually life, there was a complex process that probably went on called chemical evolution. And we're going to read a chapter in a book from The Selfish Gene called The Replicators that kind of does a really good job at explaining how this is possible, okay, and what genes are. And chemical evolution is essentially this idea that with enough energy and time, chemicals can build in complexity and can eventually form the precursors to living molecules or living things. And these molecules are the things that form life. And we have experiments that we're going to talk about a little bit later that showed that this is at least feasible, that it could have happened this way. It's like, well, how do we know the chemical evolution occurred? We don't know. We just know that we've done experiments to show it was feasible. We have fossil evidence. We weren't there at the time, so we don't know how life formed and when exactly it came about. But based on all of our models and based on all the things that we've looked at, it is possible and it is feasible. So before we get into that stuff, I'm going to talk about Louis Pasteur, or Louis Pasteur. He is the father of pasteurization, which is where we heat up milk to destroy the microorganisms in it, or other things as well. Milk's the common thing we think of, though, so that it has a longer shelf life. And back in Pasteur's days, they thought of something, that life came about by things like spontaneous generation. And they actually had recipes for making living things, like, and it sounds ridiculous now, but they would say, if you put a bunch of hay and some cheese in a pile in a barn overnight, you can spontaneously generate mice, which is ridiculous. The mice just ended up in there. But they also thought that, like, bacteria came up, like when things spoiled, they just spontaneously generated in there. And Pasteur said, no, that's probably not the case. So what he did was he would boil broth, it's like chicken broth that he got from, boiling bones and things, which has a lot of nutrients in it. He'd boil that and let it sit with the top out or the top open. And over time, you would get spoiled broth with microorganisms because they would grow in there because there's tons of food. It's a great place to live. So then what he did was he closed the top of his flask after he boiled it. And then he found that no microorganisms appeared. So he said, look, clearly they came in from the top of the bottle and then they replicated, or they reproduced at that point. And people were like, no, they need oxygen. So what did Pasteur do? He's like, all right, let's make something where we can keep the oxygen flowing, but we also can prevent organisms from going in. So he created this like S-shaped tube where oxygen could flow in from the end here, but also the microorganisms would tend to get trapped down here before they could get into the bottle. And again, what he showed was that no microorganisms showed up in here. And then as soon as he broke off the top, it spoiled. So he tried to show that spontaneous generation was not happening in the case specifically of microorganisms. Now in the 1920s, there were two scientists, one from Russia, one from Great Britain, that independently postulated that the conditions of the primitive Earth probably would have favored the idea of chemical evolution and the creation of organic compounds from inorganic precursors, just due to the environment that existed on the Earth at the time. And if you can get those organic molecules, eventually there's other feasible steps we can take to eventually get the first most primitive types of life, or life-ish type things. So the Operin-Haldane hypothesis was this idea that, again, the early Earth's conditions were so that we could create organic life, or organic molecules, not life, organic molecules from inorganic ones. So they essentially thought that abiogenesis was a testable hypothesis. So in 1953, Stanley Miller and Harold Urey decided to test this hypothesis. And what they did was they created what they think were the conditions of the early Earth. They added lightning, they added with electricity, they added a bunch of gases they thought were present on the early Earth and conditions. And they added water, and they boiled that water. All things that would be completely feasible, as I said, present on the early Earth. And they let that run for a long time. And then they collected down here, there's a trap where they collected everything that was created. And they had a sampling probe where they could actually go in and take samples of what was created. And what they ended up finding were amino acids. They found from inorganic molecules, things like, as you can see here, water, methane, ammonia, hydrogen gas, carbon monoxide. They formed amino acids, alanine, valine. But some of them are ones that we don't use, but could be precursors: aspartic acid, glutamic acid, those kind of things. So they showed that from inorganics you could create organic, and they showed the feasibility of how that would happen. The problem is that Urey Miller, both Urey and Miller, and even the other scientists at the time actually got it wrong in terms of what the early Earth's conditions were like. So what they did was they retested it later with what we currently think the early Earth's conditions are, and what they found were even more amino acids, and they also created the purines and pyrimidines that are the basis for DNA and RNA. So again, they showed that it's feasible that from inorganic molecules, we could create organic ones that are the precursors for life. They also would show that once you have amino acids, if you drop them on hot, moist clays, we've done experiments like this, we will get polypeptides. So the clay acts almost like an enzyme that allows for the synthesis of long chains of amino acids. And remember, long chains of amino acids are proteins, and proteins are the machines that allow life to exist. They essentially are what life is, right? Without proteins, you don't have life. Another cool thing is once phospholipids came about, which they could come about by natural processes as well, phospholipids will always spontaneously form in a bilayer, right? So if you put them in water, they will orient themselves with the tails pointing that are hydrophobic towards each other, the hydrophilic heads towards the watery environment, and they will actually create cell-like structures just like a cell membrane would. And you can imagine if you start trapping some of these, if there's early RNA molecules and things and proteins within this layer, it could form something similar to what a cell would be, at least early on. We also hypothesized that RNA may have been the first genetic material, right? So DNA was not the first genetic material, RNA was. And the reason they think that is because RNA is single-stranded, so it can actually copy itself because it has free bases on it. So it acts as a template to copy itself as an opposite kind of copy. And it could propagate itself that way and replicate, right? Which is one of the fundamental things life has to be able to do to exist. They also think that there's a lot of catalytic ability for RNA as well. It can act as a enzyme, essentially, called a ribozyme. So both RNA has a dual function in that way. So they think that RNA was the first genetic material, not DNA, and then DNA eventually outcompeted it because it was more stable. Here's showing you that idea that we have RNA monomers over here. Then we can get short RNA polymers, and our polymers can form again on clays and things like that. And because they're single-stranded, you can have base pairing. And when the other when the other nucleotides line up, it can copy itself. And that would be like the original gene that was copyable in early Earth, or on the early Earth. So the RNA world hypothesis, again, is the idea that the RNA was the first the first genetic material. And eventually you can see how RNA started to get attached with amino acids, which are like tRNAs. You can see how the RNAs could line up and form long chains and form proteins that way by the RNAs kind of binding to each other sideways. They're here with base pairing rules and assembling amino acids in order. Again, not exactly how life looks now, but it's a feasible way in which we can create complex molecules, and RNA helps to assemble the template for that. So what are the characteristics of the universal common ancestor of all living things? Remember, it has to be heat-loving, salt-loving, hypothermophilic, meaning like hot things. It had to be anaerobic because there was no oxygen around. They were a chemolithoautotroph, which means that they made their own food using chemicals from the earth. They could probably use hydrogen gas or sulfur compounds or something as electron donors in some sort of electron transport chain in order to do and create energy that way. It also the universal common ancestor of all living things did glycolysis. It probably had DNA at this point, and it probably had the genetic code because all living things share those characteristics, as well as ribosomes, right? You want to remember that for the AP exam. They love to ask that question. The big ones, glycolysis, genetic code, are the two biggest ones, and also ribosomes in terms of organelles. Even though ribosome depending is not technically an organelle. So where life began is another issue. So until recently, most researchers favored those shallow water and moist sediments like the clays I said, but then the discovery of deep hydrothermal vents in the 1970s created possibility that maybe it was created down there, like deep in the ocean, the first life, because it provides energy. There's also some hypotheses that we think that maybe it was extraterrestrial. And I don't mean like the aliens. I mean like life was seeded, or the molecules of life were seeded from asteroids and things, and meteors that hit the Earth. And we've tested meteors, and sometimes we actually find amino acids and things inside of the meteors and on the surface of the meteors that we know were not contaminated from the Earth itself. So it could have been seeded. It's probably a mixture of a lot of different things that eventually led us to the molecules that allowed life to develop. And remember, when I talk about these types of origins, sometimes people feel uncomfortable with that because of how they feel, because of their religious convictions and things like that. Remember, now obviously, as I said, science can sometimes be an issue with, have an issue with the fact that the Earth is 10,000 years old, right? We have a lot of evidence that shows that is not the case, but you can evoke religion or God or whatever you believe at any point in this process. God could be a being that helps to push this process forward, or created this process, right? It doesn't have to be as simple as, just reading stories as actual history and things like that. So you can do that too. For this exam, you just have to understand how this works. And even if you don't necessarily believe that it's the case, it's important to understand that how this theory works. And this tends to be the scientific consensus, is chemical evolution and how life began. So life on Earth began between, as I said, 3.5 and 4 billion years ago. The life was simple. They did glycolysis. Remember, I talked about that. It continues today. We also have the universal genetic code, which is how it reads the DNA or the RNA that turns it into protein. It's the codons in the chart that we use, right? The language. Prokaryotes then dominated Earth's history from 3.5 to 2 billion years. So nothing existed for 1.5 billion years other than things that were bacteria-like, right? Very simple. And then early on, there was a branch off between bacteria and archaea, which are those bacteria that now live in extreme environments. And they are also the bacteria that we are most closely related to. As you can see, the tree of life down here, we shared a common ancestor with archaea at this point right here. But remember, there was no oxygen yet. So there's no cellular respiration. They were just doing glycolysis early on. Oxygen started to accumulate in the atmosphere about 2.7 billion years ago, and we see this as a layer of rust in the fossil record for the first time, because rust cannot form. There's plenty of iron, but rust cannot form unless oxygen is present. So when we cut on rocks, we can see a layer of rust that really appears for the first time, meaning oxygen was present on the surface when those rocks were formed. And most of that oxygen came from photosynthesis, specifically the water splitting step of photosynthesis. Remember, what happened with photosynthesis, or in the first step of photosynthesis, is water goes in, the water is split, and electrons are liberated. They get excited to a higher energy state, and then they're passed from complex to complex, pumping hydrogens into the thylakoid. Those hydrogens flow across ATP synthase, making ATP, and then they get excited again in photosystem 1, and those electrons end up on NADPH, and that ATP and NADPH is used in the Calvin cycle to make sugar from carbon dioxide, using an enzyme called Rubisco to fix that carbon dioxide. That's basically a review of all of photosynthesis and a lot of what you need to know. Not all of it, but a lot of it. So as I said, what evolved first, photosynthesis or respiration? Photosynthesis evolved first, because that's the first time oxygen was present. And oxygen caused massive death because oxygen is an oxidizer. It rips electrons off of things. So a lot of life actually went extinct that wasn't able to adapt when oxygen began accumulating. Then some organisms started to hack that and use oxygen as that terminal electron acceptor in cellular respiration, and that has been the dominant process for energy creation, or at least energy transfer from glucose to ATP that we still use today. So there's this idea that I want you to understand called parsimony, which is also related to something called Occam's razor, which is this idea that the simplest explanation is usually the correct one. Now, the way this could work, if you actually look at the evolutionary relationships between single-celled organisms and different lineages, even of multicellular organisms, we can see photosynthesis pop up several different places. And even though these are both this is heterotrophic and this is autotrophic, and they're more closely related based on DNA evidence, we can see, oh, photosynthesis pop. This is photosynthesizing and this is not. Well, what could have happened is photosynthesis could have independently evolved. Now, it's a complex process, right? 5 separate times. Or photosynthesis evolved first, and then it was lost 3 separate times. It makes much more sense parsimoniously that it was lost 3 times, because it's much easier to lose a function than to gain a function, especially a complex process. And also there's fewer steps that have to happen in order to get that outcome. So what the prevailing theory, or hypothesis I should say, is that photosynthesis evolved initially and then it was lost in those other lineages. Eukaryotic life, so finally, like complex cells with organelles and nucleus, all that began 2.1 billion years ago. The oldest fossils of eukaryotes are 2.2 billion year old organisms that look relatively simple, like single-celled algae. This is when endosymbiosis happened, which is when the mitochondria and chloroplasts were engulfed, right, by an ancestral eukaryote. Or evidence is this circular DNA that's found in both of those organelles, also the prokaryotic like ribosomes that they have, and their multiple membranes that they have on the outside of them. And there's again showing you endosymbiosis, which we've talked about, and also the evolution of through pinching and increasing the surface area to volume ratio to increase efficiency. We see the surrounding of the nuclear of the DNA with a nucleus through pinching of the cell membrane. We can also feasibly see how the ER and the Golgi form through that process, and then we see endosymbiosis of both mitochondria and chloroplasts there. We also think that there is actually a secondary endosymbiosis between for the chloroplast, because they have 3 sets of membranes instead of just 2 like a mitochondria does. It's just showing you again kind of the loss of photosynthesis in some of these single-celled eukaryotic organisms here, as well as in fungi and animals. We can see plants branch off, multicellular plants branch off here. And then we get into animal evolution. So you can see here, here's our animals. We're kind of to this point, or excuse me, not to animal evolution, to multicellular evolution. And you can see where we are on the timeline right here. Multicellular eukaryotes didn't even exist. So things that were not microscopic didn't even exist until 1.2 billion years ago, and those were like tiny microscopic multicellular things. Larger organisms that would be visible to the naked eye, including things like jellyfish and worms, did not appear in the fossil record until only 600 million years ago, right? I know it seems like a long time, but the Earth is 4.6 billion years ago, and life showed up around, between 3 and 4 billion years ago. So multicellular life that was larger than like the head of a pin didn't exist until around 600 million years ago. You can kind of see where we are right here, multicellular, then a little higher algae, a little bit more complicated where we're at. And then finally, animals, multicellular animals didn't really exist until they existed, but the major explosion of animal diversity occurred during the Cambrian period. It's called the Cambrian explosion, when all of the current phyla of animals came about during the early Paleozoic or Cambrian period. Though some did exist, obviously, in the late Precambrian, which means before that Cambrian period, which are the cnidarians, which are the jellyfish and sponges, which are very simple multicellular organisms. Plants and fungi didn't colonize land, so they're still in the water. No land colonization occurred until 500 million years ago. And remember, in order to do that, there's a challenge. Like if you live in the water, there's a lot of things you don't have to worry about. Like you don't have to worry much about having vascular structures that provide you with the ability to deal with gravity. Because water, you don't have to worry about gravity that much because of the hydrostatic pressure on all sides of you. Once you get out of the water, you're kind of pushed down by gravity. So they have to have structures that make them have more support on land. So like think about plants, like they'd have to have roots that are really strong roots that would allow them to anchor, but also vascular structures that would allow them to be able to stand up straight and hold themselves up. They also have to stop from drying out because now you're not surrounded by water all the time. So harder external skins that are waterproof and keratinized. And keratin's a waterproofing protein that we have in our skin and nails. And also you would see that in other scaled organisms. They have to be able to reproduce outside of the water. It was so easy. You just release eggs into the water and sperm would swim to them. Well, now we have to have reproductive strategies that are more involved with like internal fertilization for things that live on land. Eggs also would dry out. So the eggs have to have harder coatings on the outside that are waterproof. There's so many things that had to happen in order for these animals to colonize, and plants for that matter, to colonize land. And this is actually called Tiktaalik, which is the first fish that they thought went on land, and it had stronger arms because it would swim around in the shallows and eat food, and it finally was able to climb up on land. They also have a swim bladder that they think became really well vascularized or filled with blood vessels that they could then diffuse oxygen into the bloodstream that way, which became like their rudimentary lungs. The first vertebrates, or jawless fish, evolved around 550 million years ago. Amphibians, which are, they're tied to the water in their juvenile stage, but then they can go on land during their adult stage, 365 million years ago. Reptiles and mammals diverged 300 million years ago, and birds diverged from dinosaurs 150 million years ago. Not that long ago in the scope of all of time. The first true primates appeared 34 million years ago. The first hominids, the great apes, about 12 to 13 million years ago. And we diverged from our closest extant alive common ancestor, the chimpanzee, about 5 to 7 million years ago. But as I said, the story doesn't end there, right? There's a lot of things that went extinct that more closely related to humans than chimpanzees, that have gone extinct. In the last 300,000 years, there were 4 different hominid species that all coexisted: Homo sapiens, Homo erectus, Homo neanderthalensis, and Homo heidelbergensis. There's also Homo florensis, which isn't even on here, which existed as recently as 10 to 20,000 years ago. And here's showing you, so there's Homo florensis. It lived on an island, so it was smaller. These are what a lot of these, we think these things look like. Here's Homo sapiens number 2. You can see Neanderthal is this one right over here. Homo habilis, which is over here. I apologize because they're hard to really see and read. There's Homo erectus, the first tool using hominid. And essentially all of these went extinct eventually, with the exception of us. Us and Neanderthal were, other than Homo florensis, were the ones that existed until only, within the last 20,000 years or so, we existed together. And then we outcompeted Neanderthal, and we actually interbred with Neanderthal. There's a lot of Neanderthal DNA in certain lineages of European populations. I hate this picture because it kind of gives a bad image of how evolution actually works, because you didn't turn from this to this, right? But this kind of gives you an idea of some of those intermediate common ancestors looked like, even though they were a distinct species that eventually led to human beings. Here's showing you some more of those evolutionary relationships. Again, that hobbit person that evolved from Homo erectus in Flores, Indonesia, until not that long ago, but did go extinct. And there's us and Neanderthal, the most closely related, even though there's that hobbit person, as I said, which is really fascinating to me that existed so close to current day. This is just a weird picture, but this is showing you what some of these early species would have looked like. Quick change. All right, so the last thing we're going to really talk about is major extinction events, and then I'm going to, at the end, talk a little bit about some other ways that, bacteria exchange DNA, just as a review of that. So So there are really 5 main mass extinction events that occurred in Earth's history that we see through the fossil record. The Ordovician, the Devonian, the Permian, the Triassic, and the KT extinction. And, you know, the one we tend to think the most about is when the dinosaurs went extinct, which was the KT extinction, right? 80% of all species went extinct in that extinction event. But the most devastating event was actually the Permian, and 95% of all species went extinct around 250 million years ago. We think that was caused by mostly volcanic activity and an increase of methane and CO2. The rapid release of those two very strong greenhouse gases, methane is very strong, caused rapid global warming and increases in temperatures, which caused oxygen to leave the oceans. And if there's no oxygen in the oceans, anything that lives there would pretty much die. And on land, everything became bone dry. So the Permian extinction is probably the most serious. It's also called, I don't know if I just said this, but the Great Dying. The KT was the one that we tend to think of in terms of the most famous one, because it was after that one that mammals kind of took over. And remember, what happens after each of these major mass extinctions is we have a lot of adaptive radiation, which is that idea that because of all the open niches or niches, you have very rapid evolution, because individual species those individuals within the species with minor differences will kind of spread out to avoid competition with each other. The sad part is that we are currently in a 6th mass extinction. It's deemed the Holocene extinction, and the Holocene is basically since the humans have been around, right? We think that due to human activity, we are destroying our environment in the ecosystems at such a rate that we are causing extinction at a rate that's 10 to 100 times higher than any of those previous mass extinctions that we have looked at. 10 to 100 times higher than an asteroid hitting the Earth, or that massive volcanic activity causing global warming and increasing climate temperatures. So we are killing things off at a rate 10 to 100 times faster than those, which is horrifying. And you can see here, now correlation does not always equal causation, but we have good reason to believe that this is the case, is that humans are causing this extinction. You can see the extinction rate in terms of the numbers of species going extinct per year. In the 1800s, it's really low, and it's not until we really, really start to expand as a species technologically. And also, in terms of just destroying the ecosystem due to that technology that we have, that you see this increase in extinctions going up pretty much with the human population. So the current rate of extinction is estimated, as I said, to be 1,000 to 10,000 times greater than the background extinction rate, because things just go extinct even when there's not something extraneous causing extinction necessarily. So we are, as I said, 1,000 to 10,000 times higher than background extinction rate. And at this rate, 30 to 50% of all species will go extinct by 2050. So in the next 26 years or so, 25 years or so. This, as I said, the most devastating extinction event in Earth's history since life began. Worse than the Permian, which is the Great Dying. So, last thing, what's the best way to determine evolutionary relationships, right? How do we determine evolutionary relationships between different things? Well, we can use DNA analysis. So for close eukaryotic relationships, we will look at differences in non-coding DNA. So if I'm going to see if I'm closely related to you or other human beings, we're going to use non-coding DNA, or the introns, because that's a little bit more free to evolve and change, because there's no selective pressure to keep those exactly the same. And again, that may be a slight oversimplification, because we're finding that introns may have a little bit more to do with phenotype than originally thought, but I digress. for looking across things like domains, like we're comparing us to bacteria, we're going to look at ribosomal RNA genes. If we want to look at things like mammals, right, we may look at other types of proteins that are important and see the differences in those proteins. So essentially the idea is that the more closely related we're going to do things that like non-coding things, the more far apart related we are, we'll start to move into proteins. And if we're really not related to these things, like with bacteria and stuff, we'll look at ribosomal RNA genes, because all things, first off, they're not barely changed at all because they're so important that they need to be conserved, but there are differences between bacterial ribosomes, archaeoribosomes, and eukaryotic ribosomes. And last thing, as I said, a review of how bacteria swap or gain DNA. Remember, there's bacterial transformation. We did a lab on that, where bacteria pick up circular pieces of DNA, that's the plasmid lab, from the environment, and can express those genes. Like antibiotic resistance, we can use those plasmids to insert genes into and make bacteria make protein products for us. There's bacterial conjugation, which is essentially when one bacteria sends a sex pilus to the other one and swaps DNA through that. They can swap plasmids and things like that. And then there's transduction, in which a bacteriophage infects a bacteria with its DNA, and instead of that, the phages that are supposed to be manufactured inside of that bacteria having DNA for the virus, they will have DNA from the bacteria, so it could pick up an antibiotic resistance gene and inject that into another bacteria, as opposed to injecting it with the viral DNA that makes it actually make copies of the virus for the virus. And here, showing you examples of each of those, there's transformation. There's the dead bacteria and another bacteria picking up the DNA, conjugation with the sex pilus and swapping of some of their DNA, and then transduction with the phage picking up, here's that one, the transducing phage as opposed to the infectious phage, injecting the gene and putting it into the cell that has now been transduced. All right, that's it. If you have any questions, feel free to ask me.